Welcome to the Deep Dive, your shortcut to being well-informed. We cut through the noise to get you those really important nuggets of knowledge.
And today, we're jumping into something really complex, but also incredibly important.
Yeah, we're tackling the century-long quest to understand and, hopefully one day conquer Alzheimer's disease.
It's a huge global health challenge. I mean, the numbers are just staggering. Back in 2020, we were looking at maybe 50 million people worldwide affected.
50 million.
Yeah.
And that's projected to go way up, right?
Way up. The forecast is nearly triple that 153 million by 2050. Think about the impact on individuals, families, entire healthcare system.
Absolutely.
And the cost.
$1 trillion annually. Globally, it's mind-boggling.
And that impact, the memory loss, the confusion, it's something so many people know firsthand or, frankly, fear. So our goal today is to trace how the science evolved.
Right, from that very first discovery through decades where, honestly, it felt like understanding was stuck.
Yeah, a real stagnation. All the way to the breakthroughs we're seeing right now. Diagnostics, therapies that actually seem to modify the disease. It's a whole new era.
And we'll look ahead, too, right, towards prevention, maybe even personalized medicine. We'll unpack how the thinking shifted, talk about the graveyard of drugs, which sounds grim, but actually taught us a lot.
It really did. Painful lessons, but crucial ones. And we'll explore the exciting new frontiers that are opening up.
So why should you care about this deep dive? Well, maybe you're prepping for a meeting, need to get up to speed on this field, or you're just curious about one of the biggest health crises we face.
Yeah, this story really highlights how science works. It's not just eureka moments, it's setbacks, it's shifts in thinking, it's connecting dots, sometimes decades apart.
Exactly. Those aha moments often come after a lot of uh-oh moments.
Yeah.
And we've dug into a lot of sources for this. Critical reviews on how the disease works, the latest scientific outlooks, clinical trial data, trying to get that full picture.
Okay, so where do we start? To really get where we are now, you have to go back, way back.
Right back to the beginning. Germany, 1901. A psychiatrist and neuropathologist named Dr. Alois Alzheimer.
He was working in Frankfurt, and he encountered this patient, Auguste
Dies. And she was relatively young, wasn't she, for the kind of symptoms she had?
That was the striking thing. She was only 50. But she had severe memory problems. She was disoriented, paranoid. Her language was affected. It was a profound cognitive decline, very unusual for her age at that time.
So Alzheimer was intrigued.
Deeply. He called it this unusual presenile dementia. He followed her case very closely, meticulously documenting her symptoms for 5 years, right up until she passed away in 1906.
And then he did something quite remarkable for the time.
He did. He asked for her brain to be sent to his lab in Munich. And in November 1906, he presented his findings. He'd examined her brain, not just looking at it, but using these new silver staining techniques under the microscope.
And what did he see?
Well, first, the brain's outer layer, the cortex, had shrunk. But the really key things were microscopic. He saw these clumps. Abnormal protein deposits outside the nerve cells, he called them senile plaques.
Okay, plaques.
And then inside the neurons, he saw these tangled bundles of fibers. We now call those neurofibrillary tangles.
Plaques and tangles,
Perfect.
the two hallmarks we still talk about today.
Exactly. Amyloid plaques and tau tangles. That was the first time anyone had directly linked a specific clinical picture of dementia to unique physical changes in the brain visible under a microscope. It was groundbreaking.
You'd think the scientific community would be buzzing.
You would think. But here's the kicker. His presentation at this big psychiatric meeting. Apparently, it was met with, well, almost complete silence.
Seriously, no questions.
Reportedly, none.
Yeah.
The story goes that the audience was more interested in the next talk, which was on, wait for it, compulsive masturbation.
You cannot be serious.
It sounds unbelievable, but it's often cited. It just shows how little traction his findings initially got. It kind of foreshadowed decades of the disease being, well, overlooked.
So it wasn't an immediate aha moment for the field.
Not at all. Though his boss, Emil Kraepelin, who was a very influential figure, he saw the significance. In his 1910 textbook, Kraepelin described the condition and actually named it Alzheimer's disease.
So it got its name, but it didn't really change how people thought about it.
Not fundamentally, no. For the next 60 years, roughly, Alzheimer's was seen as this really rare thing, something that only affected younger people, like Auguste D., that presenile category.
And if older people showed similar symptoms.
That was just written off as senility, considered a normal, inevitable part of getting old, untreatable, nothing you could really do about it.
Wow, that's a massive conceptual error, looking back.
Huge. Because it created this artificial divide, it meant that what was actually the same disease process happening in older people wasn't recognized as Alzheimer's.
So it stayed this niche, rare diagnosis.
Exactly. Which meant very little research funding, very little attention. Think about it. Between the 1910s and the early 1970s, fewer than 150 scientific papers were published on Alzheimer's, for what we now know is a leading cause of death. It's staggering.
A tiny number. So when did the light bulb finally go on? What changed that perspective?
The turning point, the real paradigm shift, came in 1976, thanks to a neurologist named Dr. Robert Katzman.
Katzman, what did he do?
He published this editorial in the Archives of Neurology, and he made a bold, simple argument. He said, look, the plaques and tangles we see in the brains of elderly people with senile dementia are identical to what Alzheimer's described in his younger patients.
Well, it's the same disease.
Exactly. He basically said, stop separating them. Presenile, senile, it's all Alzheimer's disease. And he didn't just stop there. He reframed it.
How so?
He argued it wasn't some rare curiosity. He called it a major killer, a massive public health problem. He estimated it was the fourth leading cause of death in the U.S. That completely changed the conversation.
From rare disease to major killer. That's a huge leap.
It really was. It woke everyone up, policymakers, researchers, the public. Suddenly, there was urgency. It's probably no coincidence that the National Institute on Aging, NIA, had just been founded in '74. They held their first big AD conference in '77.
And the Alzheimer's Association was founded soon after, right?
Right, 1980. So Katzman's paper really catalyzed the whole modern era of Alzheimer's research, funding, and advocacy.
So the big lesson here, it wasn't about discovering something totally new under the microscope. It was about reinterpreting what was already known.
Precisely. The plaques and tangles weren't new. The mistake was the framework, the concept. Katzman connected the dots, synthesized the evidence, and created the public health imperative that had been missing for 70 years. It shows how critical just thinking about a problem differently can be.
Okay, so Katzman opens the floodgates. Research starts ramping up. What happens next? Where did the science focus?
Well, once researchers figured out the molecular identity of those plaques and tangles in the mid-80s beta amyloid, or amorin for the plaques in '84, and the tau protein for the tangles in '86, things really started moving.
Right, they knew what the physical stuff was.
Exactly. And that led pretty quickly, in the early '90s, to the formulation of what became the dominant theory for decades, the amyloid cascade hypothesis.
The central dogma, basically. Explain that. What was the core idea?
The hypothesis, put forward by people like John Hardy and Dennis Selkoe, was elegantly simple, really. It proposed that the initial trigger, the first domino to fall in Alzheimer's, is an imbalance in the brain's handling of this A peptide.
How is A normally handled?
It's produced when a larger protein called APP gets cut by two enzymes, beta secretase and gamma secretase. Normally, it's produced and cleared away. The hypothesis said the problem starts when either too much is made or not enough is cleared out, especially a particular sticky version called A42.
42. So that specific type starts building up.
Right. And according to the hypothesis, this accumulation is the key. First, it forms these small soluble clumps called oligomers, which are thought to be really toxic to synapses, the connections between neurons.
Okay, toxic oligomers first.
Then, these oligomers clump together further to form the large, insoluble amyloid plaques that Alzheimer's originally saw. And this amyloid pathology, the hypothesis went, sets off a whole cascade of downstream events.
Like what?
Synapse dysfunction, chronic inflammation in the brain, problems with the tau protein leading to those tangles inside neurons, and ultimately widespread neuron death and dementia. It was a linear story. I starts it, everything else follows.
That sounds logical, step by step. What made it so compelling, so dominant for so long?
The genetic evidence was incredibly powerful, especially from those rare, devastating forms of early-onset familial AD or FAD.
The ones that run strongly in families and hit people young.
Exactly. Researchers found mutations in 3 specific genes that caused FAD. One was the APP gene itself, the gene for the precursor protein. The other 2 were PSEN1 and PSEN2.
And how did those genes tie back to amyloid?
Beautifully, it seemed. All the known mutations in these 3 genes directly impacted A production. Mutations in APP either increased the total amount of A made or specifically boosted the ratio of that sticky A42.
Okay, so more amyloid or more of the bad type. What about PSEN1 and PSEN2?
Turns out they code for the core part of the gamma secretase enzyme, the very enzyme that makes the final cut to release EPA. And the mutations in PSEN1 and PSE2 also tweaked gamma secretase activity to produce more FR42.
Wow. So every single known genetic cause of FAD seemed to point directly at problems with amyloid production.
Precisely. It was this incredibly neat convergence. It felt like slam dunk evidence.
Were there other genetic clues supporting this?
Yes, absolutely. Look at Down syndrome. People with Down syndrome have an extra copy of chromosome 21, which happens to be where the APP gene is located.
So they have an extra dose of the amyloid precursor protein gene.
Right. And almost invariably, people with Down syndrome develop the full brain pathology of Alzheimer's plaques and tangles by middle age. It was like a tragic natural experiment confirming the APP dose effect.
And the flip side, is there evidence that less amyloid is protective?
There is. A rare mutation found in Iceland, also in the APP gene, actually reduces AIR production. And people with that mutation are significantly protected against developing Alzheimer's. It completed the picture.
Okay, that genetic story for familial AD is really strong. But FAD is rare, less than 1% of all cases. What about the common late-onset sporadic form? Did amyloid fit there too?
That was the next crucial piece of the puzzle, and it fell into place in 1993. That's when the APOE gene was identified as the major genetic risk factor for late-onset AD.
APOE, I've heard of that one. There are different versions, right?
Exactly. 3 main versions, or alleles. APOE2, which seems to be protective. APOE3, which is the most common and considered neutral. And APOE4.
And APOE4 is the risky one.
Very much so. Having 1 copy of APOE4 increases your risk about 3 to 7 times. Having 2 copies, up to 12 times the risk. It's by far the biggest genetic risk factor for the common form of the disease.
So how does APOE4 connect back to the amyloid hypothesis? What does the APOE protein actually do?
Well, one of its key jobs in the brain is helping to clear ACE out. Think of it like a garbage truck for amyloid. And the problem is, the protein made by the APOE4 allele, APOE4, just isn't as good at this clearance job as the APOE3 or APOE2 versions.
So it links back to that ABL specifically, impaired clearance.
Exactly. It beautifully tied the biggest genetic risk factor for the most common form of AD right back to the central idea of the amyloid hypothesis.
You can see why it became so dominant. The genetics all seemed to line up perfectly. It gave researchers a clear target. Stop amyloid, stop the disease.
Absolutely. It was such a compelling narrative. The genetic evidence from FAD to Down syndrome to APOE4 all converged on amyloid. It offered this clear, linear, actionable roadmap for drug development. It really felt like the solution was within grasp.
But maybe that beautiful simplicity, that powerful narrative created a kind of tunnel vision.
Looking back, I think that's fair to say. The mistake, perhaps, wasn't seeing amyloid as important it clearly is, but maybe oversimplifying the story, especially for the sporadic form, assuming that the relatively simple genetic causes of FAD told the whole story for everyone.
Right.
This focus was so strong it created a lot of intellectual inertia, alternative ideas, other potential drivers of the disease. They just didn't get as much attention or funding for a long time, and it took, well, a lot of failure to shake that conviction.
And shake it certainly did.
Yeah.
Because despite that strong rationale, the drugs based on the amyloid hypothesis started failing dramatically, leading to that period often called the graveyard of drugs.
Yeah, it was a really tough couple of decades. Billions spent, hopes raised, and then disappointment after disappointment. And cracks started appearing in the hypothesis itself, even before the big trial failures.
Like what kind of cracks?
One of the big ones was this plaque cognition gap. Researchers doing autopsy studies kept finding brains loaded with amyloid plaques, but the person, when they were alive, had shown no signs of dementia, perfectly normal cognition.
So you could have a brain full of plaques and still be sharp. That doesn't quite fit the simple cascade idea.
Exactly. It suggested plaques alone weren't the full story, weren't sufficient to cause the symptoms. In contrast, when they looked at the amount and spread of the tau tangles, that correlated much more strongly with how severe the dementia was. People with more widespread tangles had worse cognitive decline. That led some people to think maybe tau is actually closer to the real driver of symptoms.
So the focus started to shift a bit, even within the amyloid camp.
Yeah, people started talking more about the smaller, soluble oligomers being the really toxic species, rather than just the big, inert plaques. But still, that fundamental gap, lots of plaques, no dementia, remained a puzzle.
And then came the clinical trials, an almost unbelievable string of failures.
It was rough. Huge investments, large phase 3 trials, just hitting wall after wall. They tried different approaches based on the hypothesis.
Like trying to stop A-beta production.
Right, using secretase inhibitors. First, they tried gamma secretase inhibitors, aiming to block that final enzyme cut. Drugs like semaglutide.
How did that go?
Badly. It turns out gamma secretase does a lot of other important things in the body, cutting other proteins besides APP. Blocking it caused significant side effects, like increased skin cancer risk, and actually seemed to worsen cognition in some trials. A clear failure due to off-target effects.
Okay, so gamma secretase was too broad a target. What about beta secretase, the other enzyme involved, BACE1?
That seemed more promising initially. BACE1 inhibitors like veribecestat were developed, and they did successfully lower AA levels in the brain and spinal fluid. They hit their target. But they consistently failed to slow cognitive decline in large trials. Again, some even showed hints of worsening cognition. It was deeply puzzling and a major blow to the hypothesis. If lowering AT doesn't help, what does that mean?
So stopping production didn't work. What about clearing the amyloid that's already there? Using antibodies?
That was the other major strategy. Monoclonal antibodies designed to bind to AC and help the immune system clear it away. But the first generation of these also failed.
Names like bapineuzumab, solanezumab.
Right. Bapineuzumab targeted aggregated AC in plaques, didn't show clinical benefit, and it introduced this major safety issue called ARIA, amyloid-related imaging abnormalities, basically brain swelling or microbleeds seen on MRI scans.
And solanezumab targeted the soluble single MOR molecules, right, before they clump.
Yes, the idea was to nip it in the bud. But it failed in multiple large trials in people with mild to moderate AD, and then, perhaps most tellingly, it failed in the A4 prevention trial.
Tell me about that one.
The A4 study tested solanezumab in older adults who were cognitively normal but did have amyloid buildup in their brains, confirmed by PET scans. The hope was, if you intervene really early, before symptoms, you can stop the cascade. It failed. After almost a decade, the results in 2023 showed no benefit. It strongly suggested that just targeting soluble AC without effectively clearing the established plaques wasn't enough. Other antibodies like crenezumab and gantenerumab also failed around this time. The graveyard was getting crowded.
Okay, here's where it gets really interesting. These failures, as painful and expensive as they were, ended up being the best teachers the field ever had, didn't they? What did we learn?
Absolutely crucial lessons. The first, and maybe the most important, was timing is everything. Meaning, by the time someone has even mild cognitive impairment or dementia symptoms, the disease process is likely quite advanced. Amyloid might have kicked it off years ago, but now you've got widespread tau pathology, inflammation, the damage might be self-sustaining. Trying to just remove amyloid at that late stage might be too little too late.
So the realization was you have to intervene much, much earlier, maybe even before symptoms start.
Exactly.
Yeah.
That shifted the whole focus towards preclinical intervention. Hit the disease when it's just amyloid before the other fires start raging.
Okay, lesson 1, timing. What else?
Lesson 2, treat the right patients. This sounds obvious, but
It wasn't happening.
shockingly no. Many early trials enrolled people based just on their clinical symptoms. Later, when they went back and did amyloid PET scans on stored samples or in newer trials, they found that maybe 20–30% of the participants didn't even have Alzheimer's pathology.
Wow. So they had dementia from other causes.
Right. And an anti-amyloid drug was never going to help them. Including them in the trial just diluted any potential positive signal from the true AD patients and made it harder to see if the drug was working.
So that led to
The now standard requirement. You absolutely must have biological proof of amyloid pathology, usually from a PET scan or CSF test, to get into an AD clinical trial testing an anti-amyloid drug. Screen failures went up, but the trials became much more likely to yield clear results.
Makes sense. Lesson 3.
Lesson 3 was about the drugs themselves, the need for robust target engagement, specifically the ability to actually clear the existing plaques.
So not just binding to soluble ALAC like solanezumab did.
Right. The drugs that eventually showed some success were the ones that demonstrated really profound clearance of the deposited amyloid plaques from the brain. It suggested you couldn't just stop new stuff from forming. You had to actively remove the burden that was already there.
Okay. And the final big lesson from all these failures.
It was the vindication of complexity. These failures proved, quite painfully, that Alzheimer's isn't just about amyloid, it's multifactorial.
Right. Amyloid might start the fire.
But tau pathology, neuroinflammation, vascular problems, synapse loss, these aren't just passive consequences. They become active drivers of the disease in their own right. Once they get going, they contribute significantly to the cognitive decline.
So that really opened the door wider for research into those other pathways and for the idea of needing combination therapies.
Absolutely. It forced the field to move beyond a purely amyloid-centric view and embrace the true complexity of the disease. It was a necessary, albeit difficult, evolution.
So after all that disappointment, the tide finally started to turn in the early 2020s. A new generation of anti-amyloid antibodies built on those hard lessons began to emerge. This marks the start of the modern therapeutic landscape.
Yes, and the first one out of the gate, causing a huge stir, was aducanumab, marketed as Aduhelm.
Right, approved by the FDA in 2021 under their accelerated pathway. The first new AD drug targeting the underlying pathology in nearly 2 decades. But it was incredibly controversial.
Hugely controversial. Its development involved 2 identical phase 3 trials, Emerge and Engage. Both were actually stopped early because an initial analysis suggested they were futile, unlikely to work.
Stopped early, but then it got approved.
Yes. Later, the company reanalyzed the data. They argued that in one trial, Emerge, there was a modest slowing of cognitive decline in the high-dose group. But the other trial, Engage, clearly failed. Very conflicting results.
So one maybe worked a bit, one definitely didn't. What did the FDA's advisors say?
Their own external advisory committee looked at the data and voted overwhelmingly against approving it. They felt the evidence for clinical benefit just wasn't strong enough.
Yet the FDA approved it anyway. On what basis?
On the basis that aducanumab undeniably and robustly removed amyloid plaques from the brain, the FDA used this plaque removal, a biomarker, as a surrogate endpoint, reasonably likely to predict clinical benefit, even though the direct clinical benefit data was messy.
That caused an uproar.
A massive uproar. Many scientists felt the bar for approval had been lowered too much. Plus, the drug had a very high price tag initially and significant safety concerns. About 40% of participants experienced ARIA, that brain swelling or bleeding.
So what happened with Aduhelm in the end?
It was essentially a commercial failure. Doctors were hesitant to prescribe it, insurers were reluctant to cover it without clearer proof of benefit, and patients were wary of the risks and burden. The manufacturer eventually discontinued it in early 2024.
But even as a failure, it served a purpose, didn't it?
It absolutely did. While scientifically debated and commercially unsuccessful, aducanumab was a crucial stepping stone. It proved 2 critical things. 1, that an antibody could achieve profound plaque removal, and 2, that the FDA was willing to consider plaque removal as an approvable endpoint. That really paved the way for the next drugs in line.
And the next one, lecanemab or Leqembi, felt like the real breakthrough. It got traditional FDA approval in 2023.
Yes, lecanemab felt different. Developed by Eisai and Biogen, it targets a slightly different form of amyloid protofibrils, which are these soluble clumps that are thought to be particularly toxic precursors to the big plaques.
And its big trial, Clarity AD, was much clearer.
Much clearer. A large trial, nearly 1,800 people with early symptomatic AD, mild cognitive impairment or mild dementia, all confirmed to have amyloid pathology, and the results were statistically significant across the board.
What did it show?
Over 18 months, lecanemab slowed the rate of cognitive and functional decline by 27% compared to placebo, measured on the main scale called the CDR sum of boxes. It also hit all its secondary goals related to cognition and daily living.
27%. Significant, but maybe modest in real-world terms.
That's the debate. It's statistically clear, a definite slowing, but it's not a cure. It doesn't stop or reverse the disease. But it does represent the first time a drug convincingly showed it could change the course of AD by targeting amyloid. And it achieved very significant plaque removal, too. Most patients became amyloid negative on PET scans.
What about safety? Still the ARIA issue.
Yes, ARIA remains the main concern. In Clarity AD, ARIA, the swelling, occurred in about 12.6% of lecanemab patients, though most were asymptomatic. Only 2.8% had symptoms. ARIH, microhemorrhages, was seen in 17.3%.
And the risk is higher for some people.
Much higher for people carrying the APOE4 gene, especially those with 2 copies. For them, the risk of ARIA jumped to over 32%. This led the FDA to put a black box warning on the label, recommending genetic testing and caution in APOE4 carriers.
Okay, so lecanemab broke the barrier. Then came donanemab Kisimla, approved in 2024. How is it different?
Donanemab from Eli Lilly has a really interesting, unique target. It goes after a modified form of called pyroglutamated or pyrogluau. This form is found mainly in the core of established, mature plaques.
So it targets the older, more stubborn plaques?
Exactly. And because of this, it leads to incredibly rapid and deep clearance of amyloid plaques from the brain, faster than what was seen with lecanemab or aducanumab.
And its main trial, Trailblazer ALZ-2. What were the key findings there?
Trailblazer ALZ-2 had a novel design element. It stratified patients based on their baseline level of tau pathology, measured by PET scan. This acknowledged that tau burden matters for treatment response.
Smart. So what did they find in the main group, the ones with low to medium tau?
In that group, donanemab slowed cognitive and functional decline by about 35–36% compared to placebo on the key measures. That's a slightly larger effect size than seen with lecanemab. The benefit was smaller in patients who started with high tau levels, again reinforcing that earlier intervention is better.
35% slowing.
Yeah.
And there was another interesting finding about progression.
Yes, nearly half, about 47% of the participants on donanemab in that low-medium tau group showed no clinical progression at all after 1 year, compared to only 29% in the placebo group. That's a potentially very meaningful outcome for patients.
And donanemab also had that unique treat-to-target dosing schedule. Tell us about that.
Right. This was really novel. Patients received monthly infusions, but only until their brain amyloid levels dropped below a certain threshold, indicating plaque clearance. Once they hit that target, they stopped active treatment and went on to placebo.
So not necessarily lifelong treatment.
Potentially not. Over half the participants achieved plaque clearance and stopped infusions within a year, and about 72% had stopped by 18 months. This suggests that maybe you can clear the amyloid burden and then stop, or perhaps go on to a less frequent maintenance dose. It has huge implications for reducing treatment burden, cost, and long-term safety exposure.
Speaking of safety, with that rapid plaque clearance, was ARIA more of an issue with donanemab?
It was. The potent effect came with higher ARIA rates. ARIE, swelling, occurred in 24% of donanemab patients, with 6.1% having symptoms. ARIA-H, bleeds, was seen in about 31%. And very concerningly, there were 3 deaths in the trial that were considered related to serious ARIA complications.
3 deaths.
Yeah.
That really underscored the risks involved with these powerful plaque-clearing drugs.
Absolutely. It highlights the critical need for careful patient selection, avoiding those on anticoagulants, for example, and very close monitoring with regular MRI scans, especially in the early months of treatment. These are not simple therapies.
Okay, so we have these monumental drugs, lecanemab and donanemab, finally showing they can slow the disease by removing amyloid. But that slowing is in the 27–36% range. Despite almost completely clearing plaques, the disease still progresses, just slower. This creates a kind of new plaque cognition gap, doesn't it?
It really does. It's perhaps the most important finding from these successes. Getting rid of amyloid, even profoundly, doesn't stop the disease in its tracks once symptoms have started.
So what does that tell us? What's the big takeaway?
It strongly implies that by the time someone is showing even mild cognitive impairment, other pathological processes, the tau tangles, the neuroinflammation, the synaptic loss, maybe vascular damage, are already significantly underway and are driving the clinical decline, perhaps somewhat independently of the amyloid that kicked it all off.
So clearing the amyloid helps. It slows things down, but it doesn't address those other ongoing issues.
Exactly. It validates amyloid as a crucial early target and a necessary part of the treatment strategy, especially early on. But it powerfully highlights that amyloid removal alone is insufficient, particularly once the disease is clinically manifest.
Which points directly towards the need for
combination therapies, targeting multiple pathways simultaneously or sequentially. That's clearly where the future has to go to achieve more profound clinical benefits. These first drugs are a massive step, but they're likely just the beginning.
And that naturally leads us to broaden the focus, looking beyond just amyloid. The field is now seriously exploring these other mechanisms as drivers and therapeutic targets. Let's start with tau.
Right. Remember, Alzheimer himself described both plaques and tangles. But for decades, under the amyloid cascade hypothesis, tau was seen as just a downstream consequence of amyloid. The tau hypothesis puts it much more center stage.
Remind us what tau does normally and what goes wrong in AD.
Normally, tau is a protein that helps stabilize the internal structure of neurons. Think of it like railroad ties, keeping the tracks stable for nutrient transport within the cell. In Alzheimer's, tau gets abnormally modified, specifically hyperphosphorylated.
Too many phosphate groups attached.
Exactly. This causes it to detach from the microtubules, destabilizing the neuron's transport system. And then this abnormal tau starts clumping together inside the neuron, forming those neurofibrillary tangles, or NFTs.
And that leads to the neuron malfunctioning and dying.
Pretty much. The transport system collapses, synapses fail, and eventually the cell dies. And the key evidence for the tau hypothesis is that strong correlation we mentioned earlier. The amount and location of tau tangles in the brain tracks much more closely with the severity of cognitive symptoms than amyloid plaques do.
So where there are more tangles, there's generally more cognitive impairment.
Correct. And there's further proof from other diseases.
The primary tauopathies.
Yes, diseases like certain forms of frontotemporal dementia. In these conditions, you see massive tau tangle pathology and severe dementia, but often with little or no significant amyloid plaque buildup.
So tau can clearly cause neurodegeneration and dementia all by itself, independent of amyloid.
It proves the principle, which is why developing anti-tau therapies, antibodies, inhibitors of phosphorylation or aggregation, is now a huge focus of research, seen as complementary or perhaps even primary for later stages to anti-amyloid approaches. Some researchers even talk about potential amyloid first versus tau first subtypes of AD.
Okay, so tau is a major player. What about neuroinflammation? That's moved from being seen as just a reaction to damage to being an active driver, too.
Absolutely. The neuroinflammation hypothesis has gained enormous traction, largely thanks to human genetics. When scientists did large genome-wide association studies, GWAS, looking for genes that increase risk for late-onset AD, many of the top hits weren't related to amyloid or tau directly. Instead, they were genes involved in the immune system, particularly in glial cells.
Glial cells, the brain support, and immune cells, like microglia and astrocytes.
Exactly. Genes like TREM2 and CD33, primarily expressed in microglia, popped up as significant risk factors. This strongly suggested that the brain's immune response isn't just a passive cleanup crew, it's actively involved in the disease process itself.
And these glial cells have a kind of double-edged sword role, don't they? Good cop, bad cop.
That's a great way to put it. Early on, activated microglia and astrocytes can be protective. They try to clear away all deposits, release supportive growth factors. But in the face of chronic ADOL and tau pathology, they seem to switch to a detrimental pro-inflammatory state.
So they become part of the problem.
Yes. They start releasing inflammatory molecules like cytokines, chemokines, reactive oxygen species. This chronic neuroinflammatory environment is directly toxic to neurons, damages synapses, and can actually fuel more ARO and tau pathology. It becomes a vicious, self-perpetuating cycle of damage.
So targeting neuroinflammation is another key therapeutic avenue.
Definitely. Drugs aimed at modulating microglial activation, blocking specific inflammatory pathways, are in development. The idea is to dampen the harmful chronic inflammation while hopefully preserving the beneficial functions of these cells.
Okay, cow inflammation. What about the vascular connection, the two-hit model?
This recognizes the huge overlap between cerebrovascular disease and Alzheimer's. Very few older adults have pure Alzheimer's pathology at autopsy. Most also have signs of vascular damage, like small strokes or white matter changes.
So the two-hit idea tries to link these.
Yes. The first hit is proposed to be common age-related vascular risk factors: high blood pressure, diabetes, high cholesterol, smoking, atherosclerosis. These damage the small blood vessels in the brain,
leading to reduced blood flow and,
crucially, damage to the blood-brain barrier, BBB. The BBB is this critical protective lining of the brain's blood vessels that controls what gets in and out.
Okay, so vascular risk damages the BBB. That's hit one. What's hit two?
Hit two is the consequence of that BBB damage. A leaky, damaged BBB is less efficient at clearing waste products out of the brain, including AB. So impaired clearance leads to F starting to accumulate.
So the vascular damage actually helps kickstart the amyloid problem.
That's the idea. It creates this nasty feedback loop. Vascular damage impairs AC clearance, AF builds up, and AP itself is toxic to blood vessels, causing more BBB damage, further impairing clearance, and so on. It suggests that maintaining brain vascular health could be a key way to prevent or slow AD.
Fascinating connection. Then there's the metabolic dysfunction hypothesis, the whole type 3 diabetes idea.
This one reframes AD as fundamentally a problem of energy metabolism in the brain. The brain needs a huge amount of glucose to function, but in AD, there's evidence that brain cells become inefficient at using glucose.
You can see this with brain scans, right? even early on.
Yes, PET scans measuring glucose uptake show reductions in specific brain regions years, even decades, before symptoms appear. It suggests an energy crisis is an early feature.
And the link to actual type 2 diabetes?
It's strong epidemiologically. People with type 2 diabetes have a significantly higher risk of developing AD. This led to the concept that the brain in AD might be experiencing its own form of insulin resistance, hence type 3 diabetes.
So brain cells stop responding properly to insulin, which messes up their energy supply and function.
That's the core idea. Impaired insulin signaling in the brain could disrupt neuronal function, promote inflammation, and might even directly contribute to the buildup of both ALA and tau. It's why some clinical trials are testing existing diabetes drugs like metformin or intranasal insulin to see if they can help in AD.
Okay, and perhaps the newest, most cutting-edge area is the gut-brain axis hypothesis, connecting our gut microbes to brain health.
It sounds surprising, but the evidence is mounting rapidly. This hypothesis suggests that the composition of the trillions of bacteria living in our gut, our microbiota, can influence brain health and contribute to neurodegeneration.
How would that even work? How does the gut talk to the brain?
The main proposed mechanism involves leaky gut or increased intestinal permeability. If the gut lining becomes compromised due to an unhealthy balance of microbes, dysbiosis, then bacterial components like lipopolysaccharides, LPS, which are potent inflammatory molecules from bacterial cell walls,
Well,
and potentially harmful metabolites produced by certain gut bacteria can leak out of the gut and get into the bloodstream.
creating inflammation throughout the body.
Exactly. Chronic, low-grade systemic inflammation. And this systemic inflammation can then breach the blood-brain barrier, allowing inflammatory signals into the brain, activating those glial cells we talked about, and contributing to neuroinflammation directly within the brain.
So problems in the gut could effectively fuel inflammation in the brain.
That's the hypothesis. And studies have found differences in the gut microbial composition between people with AD and healthy controls. It's a really active area of research, looking at whether modifying the gut microbiota through diet, probiotics, prebiotics, maybe even fecal transplants could be a therapeutic strategy.
So when you lay all these hypotheses out, tau, inflammation, vascular, metabolic, gut-brain, it's crystal clear they're not isolated. They all seem interconnected.
Absolutely. That's the crucial realization. The old mistake was searching for that single cause, that one silver bullet. The modern understanding is that AD is more like a network failure, a systems-level problem.
Where each part influences the others.
Right. Vascular issues worsen amyloid clearance. Amyloid and tau trigger inflammation. Inflammation damages neurons and maybe the BBB. Metabolic problems affect neuron function and might worsen pathology. Gut issues fuel inflammation. It's a complex web.
And interestingly, that major genetic risk factor, APOE4, seems to sit right in the middle of this web, doesn't it?
It really does. We know APOE4 is bad at clearing amyloid, but research also links it to exacerbated tau pathology, a heightened inflammatory response in the brain, and even effects on vascular function and lipid metabolism. It touches almost all these pathways.
Which reinforces this network idea. It's not just one thing, it's the interplay.
Yeah.
And that changes how we need to approach treatment.
Completely. It screams combination therapy. Addressing multiple nodes in this dysfunctional network simultaneously is likely necessary for maximum effect.
Okay, so while our understanding of what causes AD has become much more complex and integrated, how we diagnose it has also undergone a massive transformation, moving beyond just symptoms.
A huge shift. For decades, AD was primarily a clinical diagnosis based on symptoms, with definitive confirmation only possible after death by looking at the brain tissue. Now, it's increasingly defined biologically by the presence of those core pathologies, amyloid and tau, even in living people.
Regardless of whether they have symptoms yet.
Exactly. This was formalized in the 2018 NIAAA research framework using the ATN system. You classify someone based on biomarkers for amyloid A, tau T, and neurodegeneration N. This acknowledges that long preclinical phase where the biology is changing years before memory problems start.
And the latest diagnostic criteria update in 2024 took another big step.
A massive step. The Alzheimer's Association's 2024 criteria officially integrated blood-based biomarkers into the diagnostic process. This is huge for making biological diagnosis more accessible.
This biological definition really relied on the development of tools to see the pathology in living brains, right? Starting with PAT scans.
Absolutely. Amyloid PET imaging, which came online around 2004, was revolutionary. Using special radioactive tracers that bind to amyloid plaques, doctors could finally see and confirm the presence of amyloid pathology in a living person's brain.
Which was critical for those clinical trials we discussed.
Yeah. Essential. It ensured they were enrolling people who actually had AD pathology, making the trials much more reliable. And recently, in 2023, Medicare in the U.S. expanded coverage for amyloid PT, making it more available for clinical diagnosis too.
And then came tau PET scans, just as important.
Arguably even more important clinically because, as we said, tau tangle burden tracks much more closely with cognitive symptoms and how severe the disease is. Tau PET allows doctors to see the extent and location of tau pathology.
Which tells you more about the stage of the disease.
Exactly. It helps stage the disease, predict how quickly someone might decline, and it's also really useful for differentiating AD from other dementias that might look similar symptomatically but don't have the same tau patterns.
And the timing difference is key too, right? Amyloid builds up early, then tau.
Generally, yes. Amyloid plaques start accumulating maybe 10, 15, even 20 years before symptoms, and the rate of accumulation might slow down later. Tau tangles tend to start accumulating closer to when symptoms begin, and their spread through the brain correlates well with the progression of cognitive decline. Seeing both gives a much fuller picture.
Okay, PT scans are powerful but expensive and not available everywhere. That brings us to the real game changer. The blood test revolution.
This is arguably the most transformative diagnostic advance in recent years. The ability to detect AD pathology accurately using a simple blood test is poised to completely change how we screen for, diagnose, and manage the disease.
What specific biomarker in the blood is the star player here?
The clear frontrunner is a specific form of phosphorylated tau called p-tau217. Using these incredibly sensitive new technologies like Simoa, which can basically detect single molecules, researchers can now reliably measure tiny amounts of brain-derived p-tau217 that get into the bloodstream.
And how accurate are these p-tau217 blood tests?
Astonishingly accurate. Multiple large studies have shown that plasma p-tau217 levels can identify whether someone has amyloid plaques in their brain with accuracy that's comparable to, or sometimes even better than, amyloid PT scans, or testing the cerebrospinal fluid, CSF, obtained via lumbar puncture. We're talking accuracy often over 90%.
Wow. As good as a brain scan or spinal tap from a blood draw.
Pretty much. And it's not just good at detecting amyloid, it's also very good at distinguishing Alzheimer's disease from other neurodegenerative conditions. Plus, p-tau217 levels start to rise very early when amyloid first starts accumulating, meaning it can potentially detect preclinical AD years before symptoms.
So what does this mean for you, the listener? What's the real-world impact going to be?
It's potentially enormous. Diagnosis could move from specialized neurology centers requiring expensive scans or invasive procedures into the primary care setting. Your family doctor might eventually be able to order a blood test to screen for AD risk.
Making early detection much more widespread and accessible.
Exactly. It could dramatically speed up diagnosis, allowing people to access treatments like lecanemab or donanemab much earlier when they're likely to be most effective. It will make recruiting for clinical trials vastly easier and cheaper. And it gives us a tool to monitor disease progression or response to treatment over time with repeated blood tests.
And this isn't just theoretical anymore. It's being incorporated into official guidelines.
Yes, those 2024 Alzheimer's Association criteria designated accurate plasma biomarker tests, like p-tau217, as core 1 biomarkers. That means a positive result from a reliable blood test can now be sufficient on its own for making a biological diagnosis of AD. It's moving into standard practice.
But this incredible diagnostic power creates a huge new challenge, doesn't it?
A profound one. We're suddenly gaining the ability to tell potentially millions of people who feel perfectly healthy, who have no cognitive symptoms, that they have the underlying brain changes of Alzheimer's disease brewing.
And we don't yet have widely available, perfectly safe, and completely effective treatments to offer everyone at that stage.
Precisely. The mistake of the next decade, as some have put it, would be to perfect diagnosis without having equally robust interventions. We risk creating a situation where we can give people devastating news decades early, but without clear, accessible solutions. It puts immense pressure on developing effective preventive strategies.
Which leads us perfectly into the future outlook. The focus is shifting massively towards prevention and also towards more personalized precision medicine approaches.
Absolutely. The clear consensus now is that the best time to fight Alzheimer's is before it really takes hold during that long preclinical window. And this involved two main strategies: primary and secondary prevention.
Primary prevention is about reducing risk in the first place, right? Lifestyle stuff.
Exactly. Trying to stop the disease process from even starting. That landmark 2020 Lancet Commission report was eye-opening. It suggested that modifying 12 key risk factors could potentially prevent or delay up to 40% of dementia cases worldwide.
40%. What kind of factors are we talking about?
Things like managing high blood pressure, increasing physical activity, avoiding obesity and smoking, treating diabetes, but also addressing hearing loss in midlife, reducing depression, getting enough education, maintaining social connections, limiting excessive alcohol, and avoiding head injuries and air pollution.
So a lot of things related to general health and brain health. Are these being tested formally?
Yes, large trials like the U.S. Pointer study are actively testing whether intervening across multiple lifestyle domains simultaneously can protect cognitive function in older adults who are at increased risk.
Okay, that's primary prevention. What about secondary prevention?
Secondary prevention targets people who already have the underlying Alzheimer's biology, the plaques, maybe early tangles, confirmed by biomarkers, but who don't have any clinical symptoms yet. The goal is to intervene with a drug or therapy to stop or significantly slow the progression before cognition declines.
Like using those new antibody drugs much earlier.
Exactly. Trials like the AHEAD 345 study are testing lecanemab in preclinical individuals. The DIAN-2 study is testing drugs in people with genetic mutations for early onset AD, often starting treatment years before their expected symptom onset.
And we mentioned some early promising hints from one of those genetic studies.
Right. A preliminary analysis from a long-term observational study following the DIAN-2 trial of gantenerumab suggested that participants who received the drug for several years before symptoms were expected actually had about a 50% lower risk of progressing to dementia compared to what would be expected.
Cutting the risk in half. That's the first real glimmer of hope from a trial that prevention might actually be possible.
It's tantalizing, though still early days and needs confirmation, but it fuels the hope that intervening in the preclinical stage could be truly transformative.
Now, given everything we've said about the complexity, amyloid, tau, inflammation, vascular issues all playing a role, it seems unlikely that targeting just one thing will be enough, especially once the disease is established.
Highly unlikely. The future really has to be in combination therapy. Just like we treat cancer or HIV with drug cocktails that hit multiple targets, we'll almost certainly need to do the same for Alzheimer's.
So mixing and matching.
Yes. Maybe combining an anti-amyloid drug with an anti-tau drug, or pairing an amyloid clearer with something that dampens neuroinflammation, or adding a drug that improves vascular health or brain energy metabolism.
Could these be given together or in sequence?
Both are possible. Maybe you start with a powerful amyloid-clearing antibody to debulk the plaques, and then follow up with a maintenance therapy targeting tau or inflammation. Or perhaps, for some patients, a simultaneous multi-target approach from the beginning will be best. The key is attacking that complex network from multiple angles.
Which leads directly to the idea of precision medicine, moving away from treating Alzheimer's as one single entity.
Exactly. We need to get much more personalized, and biomarkers will be the key to this.
How so? Biomarker-guided treatment.
Yes. In the future, a patient's treatment plan won't just be based on their symptoms, but on their specific biological profile. Do they have high amyloid but low tau? Maybe anti-amyloid monotherapy is best. Do they have high levels of amyloid, tau, and inflammatory markers? They probably need a combination targeting all three. We'll tailor the treatment to the individual's specific pathology mix.
And genetics will play a role too, beyond just APOE4 for risk assessment.
Absolutely. We're already seeing pharmacogenomics with APOE4 genotyping guiding ARIA risk management for lecanemab and donanemab. Future therapies might be developed that specifically counteract the effects of APOE4, or perhaps some drugs will work better in non-carriers. We'll match the drug to the patient's genetic makeup as well.
And making sense of all this incredibly complex data, genetics, proteomics, imaging biomarkers, clinical information that sounds like a job for.
Artificial intelligence, AI, and machine learning are going to be indispensable. They can analyze these vast, multidimensional data sets in ways humans can't.
To do what?
To identify hidden patterns, discover novel disease subtypes we don't even recognize yet, predict an individual's likely disease course with much greater accuracy, and ultimately help clinicians select the optimal combination of therapies for that specific person at that specific time. AI is essential to unlocking true precision medicine for AD.
So when you trace this whole century-long story, From Dr. Alzheimer, seeing plaques and tangles in one patient, through the decades of obscurity, the rise and fall of the amyloid hypothesis, the hard lessons, the new drugs, the biomarker revolution, it feels like we're arriving at a fundamental rethinking of Alzheimer's disease itself.
I think that's exactly right. The entire journey points towards a reconceptualization.
It raises an important question, doesn't it? Will the ultimate success story be finding a single cure for Alzheimer's? Or will we be realizing that Alzheimer's isn't one thing and developing cures for, well, the Alzheimer's?
That's a brilliant way to put it. It seems increasingly likely that the singular concept is dissolving. Alzheimer's disease is probably a clinical syndrome, a common end-stage pathway that can be reached through several different underlying biological routes, or endotypes.
Each potentially requiring a different approach.
Precisely. Some people's disease might be primarily driven by amyloid dysregulation, others by inflammation, others by vascular issues, or tau, or some complex mix. The future likely involves diagnosing these specific subtypes and deploying a portfolio of targeted interventions matched to the underlying biology. Not one cure, but many tailored cures.
What an incredible arc. From this forgotten, peculiar malady that no one paid attention to a recognized global crisis. Through the dominance of one powerful idea, the amyloid hypothesis, and the painful but necessary process of learning from its limitations through trial failures.
And arriving now at these first real disease-modifying treatments, fueled by an explosion in diagnostic tools like PET scans and blood tests that let us see the disease earlier and more clearly than ever before.
And looking ahead to this really exciting, but also complex future prevention, combination therapies, and truly personalized precision medicine. It shows how science, even when it seems stuck or goes down wrong paths, eventually self-corrects and moves forward.
It does. The journey has been long, often frustrating, and involved immense resources and dedication. But the progress in just the last 5 to 10 years has been breathtaking, transforming the field from one of near despair to one of tangible hope and rapid innovation.
So here's a final thought for you, our listeners. As we get better and better at predicting Alzheimer's, maybe decades before symptoms even start, and as we develop treatments that are tailored to each person's unique biology, what does that really mean for us? How does it change how we think about health, about aging, even about our own identity, knowing this information potentially decades in advance?
It's a huge question. How do we navigate that knowledge, not just medically, but ethically, socially, emotionally? When does disease begin? When does risk become reality? These are societal conversations we absolutely need to have as the science continues to accelerate.
Definitely food for thought. This field is moving incredibly fast, so we encourage you to keep learning, stay informed, as this progress reshapes our understanding of brain health and disease.
Thank you for joining us for this deep dive.
Yes, thank you for tuning in.